Hello everyone, good day. This is Ma'am Honey and today we will be talking about enzymes. At the end of this presentation, you will be able to first describe enzymes, second describe the components of enzymes, and third determine how factors such as pH, temperature, and substrate affect enzyme activity. First is let us define the word enzyme. What are enzymes? Enzymes are the catalysts for biochemical reactions that happen in living organisms. In humans, we are all aware that there are a lot of chemical processes that are happening in our system. And in order to speed up these processes or biochemical reactions, we have this what we call enzymes, which are catalysts. And catalysts are defined as substances that speed up a chemical reaction without being a reactant. How does enzymes speed up the reaction? Enzymes speed up the reaction by lowering the activation level of the reaction. And if we say activation level, it refers to the amount of energy that must be needed in order for the reaction to occur. If we are going to look at the graph, the green curve refers to the activation energy of catalyzed reaction, while the blue curve refers to the activation energy of uncatalyzed reaction. The green curve is catalyzed, meaning it has an enzyme, while the blue curve is not. With the presence of an enzyme as a catalyst, it lowers the activation energy of the reaction, while the reaction with no enzyme or catalyst requires more energy for the reaction to be activated. How do enzymes work? Enzymes work by binding to reactant molecules or substrate. Again, the role of the enzyme is to speed up the reaction. There are reactions where one substrate is broken down into multiple products, while there are also reactions wherein two substrates come together to create one larger molecule. Again, the role of the enzyme is to speed up these reactions. Now, how does it work? The part of the enzyme where substrates bind is called the active site. When the substrates are binded to the active site of the enzyme, an enzyme-substrate complex will be formed. After the formation of the enzyme-substrate complex, the enzyme will then speed up the reaction. After the reaction, an enzyme product complex will be formed. And when the enzyme is done catalyzing the reaction, it will just release the product and will be ready again for the new cycle. Enzymes are very specific. Enzymes are usually proteins, and proteins are made up of amino acids. The active site of an enzyme gets its properties from the amino acid. These amino acids may have side chains that are large or small, acidic or basic, or hydrophobic or hydrophilic. With that, the amino acids found on the active sites of these enzymes give them the very specific size, shape, and chemical behavior. Meaning, enzymes or the active sites of these enzymes are uniquely suited to bind a particular substrate. Again, enzymes are very specific and only worked with a certain substrate. Next is the enzyme and substrate relationship. Enzymes do have the ability to change slightly when they bind substrates in order to have an even tighter fit. And it is what we called induced fit. If we are going to look at the illustration, the first image of the enzyme and substrate doesn't perfectly match with each other. After the substrate binded to the active site, the enzyme changed its shape to have an even tighter fit with the substrate. Knowing how enzymes work, now let us proceed to the factors that could affect enzyme activities. First is temperature. How does temperature affect the enzyme activity? The chemical bonds in the active sites of enzymes will be affected if the temperature is above or below the optimum temperature or the most conducive temperature of the enzyme. If it's above or below the optimum temperature, it will cause the enzyme to be less suited to bind substrate. If we are going to look at the graph, the optimum temperature of the enzyme is 40 degrees. And if it is below 40 degrees, it will cause the molecules found in the active site to gain energy. And if it's above the optimum temperature, it will cause the enzyme to be denatured, or it could alter the enzyme's natural properties or qualities. With that, it will cause the enzyme to be less suited in binding substrate. Another factor that could affect the enzyme activity is the substrate concentration. Enzymatic activity is directly proportional to the substrate concentration, meaning if the substrate concentration increases, the enzyme activity will also increase until the limiting rate is reached. After it reached to its limiting rate, further increase of substrate concentration will no longer affect the reaction rate. If we are going to look at the graph, at low substrate concentration, only one substrate is attached to an active site out of 3 enzymes present. At medium substrate concentration, the enzyme's activity also increased, where 2 substrates are attached to the active sites out of 3 enzymes present. At high substrate concentration, the limiting rate is reached with the presence of excessive substrate, or the enzymes are saturated with substrate. With that, all enzymes' active sites have substrates bound to them. Another factor that could affect enzyme activity is the pH. pH refers to the acidity or basicity of a solution. Same with the temperature. If the pH is above or below its optimum range, it will cause the enzymes to be denatured. Again, enzymes work best with its optimum pH. The factors that could affect enzymatic activities are the temperature, the substrate concentration, and pH. Thank you for watching this presentation, and I hope that you learned something. To learn more, kindly read the contents, watch the videos, and do the activities below. Thank you and have a nice day ahead.